Hi everyone, this is Dr. Mamta Chava, infectious diseases specialist and microbiology faculty at DAMS. In this video module, I'm going to discuss an important clinical vignette on infectious diseases which was asked in the recently held INI-CT exam in November 2020. So let us start. The question which was asked was like this. A male patient presented with a complaint of urethral discharge. The urethral discharge was collected and subjected to gram stain. The findings observed are shown in the image. What organism is implicated in this case? The options are human papillomavirus, Neisseria gonorrhoeae, mycoplasma or chlamydia. Now guys, if you know the basics of staining and the basics in microbiology, you can easily answer this clinical vignette. Now let us see. This is a gram stain image and what you can see over here, you can see a lot of neutrophils. This image is studded with neutrophils and you can see some tiny dots over here which are present inside the neutrophils and outside also, and they are reddish in color, so they are gram negative and they are more or less spherical. You cannot really comment on the shape, but they're looking like dots, so they're looking like cocci. Okay, remember all the cocci are not spherical, but I think this much info is enough in this question that you are seeing something gram negative, which is definitely not a rod, and they're very tiny and they're looking like spheres, so they are gram negative cocci and they are present both inside and outside the neutrophils. Now option A is human papillomavirus. Can you see viruses in gram stain? No. So very simple to rule out. Option A is not the answer. Now let us see option B. Neisseria gonorrhoeae, also known as gonococcus. Could this be the answer? Yes, it could be the answer. It's a gram negative coccus and it is a very well known cause of urethritis, and this patient was presenting with the complaint of urethral discharge. Then mycoplasma. Certain species of mycoplasma can also cause urethritis, but can we see mycoplasma on gram stain? No guys, we can't. Mycoplasma is the only bacteria in the entire bacteriology which does not have a cell wall. And as far as gram stain is concerned, we have got several theories to explain the findings on gram stain, the gram stain reaction, gram positive, gram negative. But the most widely accepted theory is the cell wall theory that categorizes organisms into gram positive and gram negative. And mycoplasma does not have a cell wall. It cannot be seen on gram stain. We resort to other stains like GMSA stain, etc. to see mycoplasma. So this automatically is ruled out. Then lastly, chlamydia. Can chlamydia cause urethritis? Yes. There are certain serotypes of chlamydia trachomatis which can cause urethritis, but can you see it on gram stain, this organism? No, you can't, because chlamydia are obligate intracellular organisms. Chlamydia produce what are known as inclusion bodies, which we can see by certain staining techniques by subjecting the clinical specimens to certain staining techniques like GMSA stain, iodine stain, and direct fluorescent antibody stain, but not gram stain. So keeping all these basics about stains and little facts about microbes, that mycoplasma, for instance, do not have a cell wall, you can easily arrive at the correct answer, and that is option B, that is Neisseria gonorrhoeae or gonococcus. Now, the patient in this question was presenting with the complaints of urethral discharge. This is a case of urethritis caused by gonococcus, so it's gonococcal urethritis. Okay. Now, I would like to tell you some important features of gonococcus which you need to remember as medicos. So what you can see over here, first and foremost, you need to know what is the morphology of gonococci. Now remember, in the entire bacteriology, the only medically important gram-negative cocci belong to two genera, Neisseria and Moraxella. All right, and under Neisseria genus, we have got two important pathogens: Neisseria meningitidis, also known as meningococcus, and Neisseria gonorrhoeae, also known as gonococcus. Now, Neisseria gonorrhoeae or gonococcus looks like this. You can see these two evil-looking organisms. Look at their eyes. They are they have got cunning eyes. Obviously, all microorganisms, all pathogenic microorganisms are cunning only, and these fellows are even more cunning because they entail negativity. Remember, they are gram negative. That is why I've drawn them in red color. So they are gram negative. They exist in pairs. Hum saath saath hain. We are together. Okay. They are partners in crime. So these are gram negative diplococci. They exist in pairs. Diplo means two. Cocci means cocci. Now the gram stain picture which was given in the question, in that the size of the cocci was very tiny. Remember, these are very tiny cocci, maximum zero point 0.3 to zero point 0.4 microns in size. And in the picture which we had in the question, you really could not comment upon the shape of the cocci. Remember, gonococci are kidney or bean shaped, but the picture in the question, let me go back to the question once again. Now guys, in this picture, you really can't comment on the kidney shape of the gonococci. Okay. So anyways, remember that the precise shape of gonococci is kidney or bean shaped. And remember, what are these? This is not just decoration I have drawn. This is actually pili, very, very important virulence factors of gonococci. Remember, if you remove the pili from a gonococcus, it becomes avirulent. As simple as that. No pili, no disease. So pileated ones are virulent and apileated gonococci are non-virulent. Why pili are so important? Because pili are the structures which help gonococci in attaching to the host cells, okay, in attaching to the host cells. And remember, pili show antigenic variation, very, very important. Pili show antigenic variation, and based on the amount of distribution of pili on the surface of a gonococcus, you can do classification of gonococci, and that classification is known as Kellogg's typing. Khane wala Kellogg's typing. Okay, remember it like that. And gonococci are both intra and extracellular, as I had shown while telling you the question, that you could see tiny, tiny gram negative dots both inside and outside the neutrophils. So on purpose, I have drawn 1 foot inside this crime neutrophil and 1 foot outside. Okay. Now this is very important to remember, because around 2 years back, there was another question in AIMS on gonococci, where one option was whether these are obligate intracellular or not. Right now, guys, this is very, very logical that you can grow gonococci in the lab on several culture media like chocolate agar, modified Thayer Martin medium. So if at all you are able to grow an organism on artificial culture media, it cannot be obligate intracellular. And anyways, the picture which I had shown you in this question, it had cocci both inside and outside the cells. So remember, these organisms, gonococci, they are not obligate intracellular. Okay, you can grow them in the lab on artificial media, and they are motile, and the kind of motility is twitching motility. Now this is also very important. The AIMS question which I was talking about, it had got another option whether gonococci are motile or not. Now guys, this was a very good question, I would say, asked in AIMS. Because most of the books mention that gonococci are non-motile, but certain standard textbooks like Sherris Microbiology, they have commented on motility of gonococci, and that is twitching motility. So on purpose, I have drawn legs as well to help you remember that these are motile cocci, and what kind of motility they show? It is twitching motility. So remember all these important facts. Now guys, over here, I want all of you to appreciate this gonococcus trying to attach to the host cells with the help of pili. With the help of pili. In the inset, you can see this reddish picture. This shows a closer view of the gonococci, and the black and white picture at the bottom is a cross section of this colony, of this micro colony, which is shown in the inset. So you can see over here, this is the gonococcus, and these are the pili which are helping it to attach to the host cells. Okay. And this attack, basically the pili are used by gonococci to attain motility using a contractile force. So this is a peculiar feature of gonococci. Remember this. Then this is a very good electron microscopic picture of gonococci. You can see existence in pairs with beautiful pili flaunted on the surface. Then what gonococcus does to humans? Now Remember, gonococcus is unique in a number of ways. I have told you that it is a coccus which is motile, twitching motility. It exists in pairs. It is not obligate intracellular. And remember, gonococcus is an obligate human pathogen. It cannot be grown in animals. So it's an exception to Koch's postulates in that regard. So what it does to humans, it causes a sexually transmitted infection, an STI called as gonorrhea. In males, it causes urethritis, which is characterized by urethral discharge, which was the case with the patient in this question, in this clinical vignette. All right, and males are usually symptomatic. In females, it causes cervicitis, but females endocervicitis, but females are usually asymptomatic and they serve as reservoirs of infection. So in females, endocervicitis. Remember, vaginitis does not occur because stratified squamous epithelium, which lines the vagina, is resistant to infection by gonococci. So this is very, very important. Now, males and females can land up in complications because of infection by gonococci. And what are these complications? If we talk about males, urethritis can be followed by formation of periurethral abscess, and if you do not treat in time, the periurethral abscess could be followed by stricture formation, and once stricture forms, you might have to go in for surgical correction. That is one complication. Then the second complication is water can perineum, which means multiple discharging sinuses in the perineal region. Then the third complication which could happen is disseminated gonococcal infection, wherein the gonococci are literally swimming in the bloodstream. They can go here and there. They can cause systemic manifestations, and the most important ones are, remember, TRS. Tenosynovitis, rash and septic arthritis. I repeat, TRS, tenosynovitis, rash and septic arthritis. Remember, gonococci are the most common cause of septic arthritis in sexually active adults, both males and females. And this complication, disseminated gonococcal infection, is seen not just in males but also in females. Okay, so one complication common to males and females is disseminated gonococcal infection, that is DGI. And remember, DGI is more common in those individuals who are deficient in the late complement proteins. All right. Then other complications in females could be from the endocervical region. The infection could involve the fallopian tubes, that is pelvic inflammatory disease. It's a very important cause of acute PID. All right. Then PID, in case of PID, if the tubes are partially blocked, it could lead to ectopic pregnancy, and if the tubes are fully blocked, it could lead to secondary infertility. So these are the complications which could occur as a result of PID. Okay. Then the infection could spill over into the peritoneal cavity from the fallopian tubes, which we call as Fitz-Hugh-Curtis syndrome, which is characterized by perihepatitis and periappendicitis, that is inflammation of the serosal lining around the liver and the appendix. Okay, and if such a female gives birth to a baby, the baby could also acquire the infection, which we call as ophthalmia neonatorum. Okay. So this all you need to remember. And this is a picture of the urethral discharge of gonorrhea. I want you all to appreciate how purulent the discharge is. Okay. Now the discharge in most cases is purulent, but in certain cases it could be mucopurulent also. Now over there, I want to tell you that if suppose a male patient comes to you and with the complaint of urethral discharge, and you decide to do gram stain, but you are able to see a lot of neutrophils, but you can't see any organism. So what could you be dealing with? It could be a case of non-gonococcal urethritis. Now the most common cause of non-gonococcal urethritis or NGU is chlamydia trachomatis serotypes D2K. And remember, the urethral discharge in chlamydial chlamydial urethritis is mucopurulent. Remember, the urethritis caused by chlamydia trachomatis is less severe and is less commonly followed by complications as compared to urethritis caused by gonococcus. The complications which I have told you regarding the gonococcal urethritis, all those complications could be encountered in case of non-gonococcal urethritis also, but the complications are less commonly encountered in chlamydial urethritis, except one, that is Fitz-Hugh-Curtis syndrome. Remember, the most common cause of Fitz-Hugh-Curtis syndrome is chlamydia. C for common, C for chlamydia. Okay. Other than this, all the other complications which I have told you, they are common for both gonococcal and non-gonococcal urethritis, but they are more commonly encountered in case of gonococcal urethritis. Okay, so it's more severe. And this is gonococcal ophthalmia neonatorum. Okay, so this picture shows you inflammation and edema appearing 2 to 5 days after birth. Okay, and as I already said, these symptoms are more severe than those observed with chlamydial infections. Okay, so a good thing is that NGU, that is non-gonococcal urethritis per se, is more common. It is it is more common as compared to gonococcal urethritis. But as far as complications are concerned, complications are more common in case of gonococcal urethritis. Then over here, I want you all to appreciate the difference in the colony morphology which we get on the chocolate agar, which is shown on the left, and on modified Thayer-Martin medium, which is shown on the right. Now, remember, chocolate agar and modified Thayer-Martin medium, these two culture media reserved for the are used for the growth of Neisseria organisms, both gonococcus and meningococcus. All right. Now, in case of gonococcus, if you're suspecting gonococcal infections, what samples do we collect? We collect urethral discharge in case of males. We collect endocervical swab in case of females. Okay. So these samples are by default will be containing the commensal organisms, and that is why it is better to use modified Thayer-Martin medium, which is shown on the right, because if you use chocolate agar, the commensals will also grow. So on the left, you can see this culture of cervical sample on plain chocolate agar permitting the growth of contaminating organisms. Are you interested in growing commensals? No, of course not. So commensals are like traffic. You don't want to entertain the traffic. So go for such a medium which has got which can remove the commensals. For instance, in this case, modified Thayer-Martin medium. Now, why it is preferable? Because it has got these antibiotics, vancomycin, colistin, trimethoprim, and nystatin, which allow selective growth of Neisseria. How? Remember, vancomycin will inhibit the growth of gram-positive cocci in gram-positive organisms, which are members of the commensal flora. Colistin will inhibit the gram-negative commensals, but it will spare Neisseria. Then trimethoprim will prevent swarming by Proteus, and nystatin is antifungal. Okay, so if you are inoculating the samples on modified Thayer-Martin medium, then exclusively you will get the growth of Neisseria which you want. All right. Then another important characteristic, another important lab test which we do for identifying gonococci or Neisseria gonorrhoeae is the sugar utilization test. So we have got these nice liquid media called as sugar utilization media. We have got different these are liquid media, by the way, and we have got different sugars: glucose, maltose, sucrose. We inoculate or add the colonies in these media. The original color of these media is pink. Okay, if at all the organism is able to utilize the sugar, the color will turn to yellow from pink. So in case of Neisseria gonorrhoeae or gonococcus, it is only the glucose sugar which is utilized and not maltose or sucrose. Okay, so remember this: G for gonococcus, G for glucose only. Okay. So this is another important lab test. Now, as far as treatment is concerned, the drug of choice is ceftriaxone. Unfortunately, we don't have vaccine for gonococcal infections, which is logical because, as I said before, pili, which are a very important virulence factor of gonococci, they show antigenic variation. And guys, whatever organisms show antigenic variation, meaning वो गिरगिट की तरह अपना रंग बदलते हैं, they keep on changing the structure of their, they keep on changing the structure of their molecules. It could be pili or anything else. So it's very difficult to manufacture vaccines of such organisms. Okay. Then this is about this clinical vignette, and the purpose of me picking up this very question for discussion is the fact that similar question with exactly same image I have discussed in DAMS infectious disease course, which is there in the extra edge course section on the eMedicos app. The case number 10, this has been brought to my notice by students only. Okay, the same image which was asked in the recent INI-CT exam, the same image I have discussed. Okay, and gonococcus, by the way, is a very favorite this thing organism of AIMS people, and that is why I had labeled this very question as AIMS Pataka question. AIMS Pataka question. So do watch the DID series on eMedicos. Okay, there are 3 microbiology videos with clinical vignettes. Okay, so they have the questions have been chosen keeping in mind the needs of the central institutes. Okay, so do not miss out on DID also. And I would appreciate your feedback on this video, and looking forward to another video very, very soon. Thank you.